So 8.1 is really a continuation of what we met earlier when we looked at this idea of enzymes and enzymes being involved in making small intermediate steps. Also that enzymes work on an induced fit model which lowers the activation energy, etc. But in 8.1, this is a higher level unit, so you must make sure that you've got the detail to your fingertips. The essential idea here is that any chemical reaction that takes place in the cell needs to be efficient. So what we want to do is make sure that we don't want to produce too much product at any given time, because that would be energetically wasteful and use up too many resources perhaps. What we can see in this diagram here is the idea that the enzyme, it has a very, very specific three-dimensional shape. Remember that the substrate is complementary to the shape of the active site. And not only is it complementary in terms of the shape, the stereospecific shape, it's also complementary in terms of the chemistry. And what happens is when we get the right enzyme which fits into the right active site, we get an enzyme substrate complex. And when we have an enzyme substrate complex, what happens is that the enzyme goes through some sort of conformational change, which changes the bonding structure, places strain, if you will, on the substrate. And the effect of that is that we have a so-called transitional state. And the transitional state means that we are going to lower the activation energy which is required for the product to be released. Okay, and here we've got the product. Now, what this is also showing is that we've got a pathway. So we've got a number of enzymes making very, very small changes in small steps with little intermediate forms. Now, the idea here is that that allows us to perhaps control at this stage, control also at this stage, control at this stage, etc., etc. So the more steps there are, the more opportunity that we've got to control how these enzymes are performing. In addition to that, if we make changes in small steps, then we're going to produce less heat, and we don't want to produce excessive amounts of heat. So if we went from this substrate here all the way to this end product in one go, that would likely result in a lot of heat being produced, which can obviously damage any kind of protein within the cell, the enzyme included. So the essential idea is that metabolic reactions are controlled, they are regulated. And the idea of the regulation is so that we don't produce too much product in too quick a time because we don't need it. We don't want to be wasteful. We don't want to be energetically wasteful either. And we don't want to run out necessarily of substrate. Now, what you can also see here is the idea of how the end product itself can change the rate of the chemical reactions that take place here. So this end product has got the capacity to fit also within the enzyme. And this is fitting not at the active site, but a different site. This different site is referred to as the allosteric site. And when this end product fits with the allosteric site, again, it has the capacity to change the shape of the enzyme. And in so doing, it changes the shape of this active site so that the active site is no longer complementary to the substrate. So if we have a buildup of this end product, then we have lots of end product regulating the effect of this enzyme. And what I need to say here as well is that the end product can fit into this allosteric site and also come out of the allosteric site. It doesn't actually react with the enzyme per se, so it stays as one particular product. Now, that means that if we are inhibiting using this idea, we've got end product inhibition, the end product fitting with the allosteric site, preventing this first step from taking place. And if the first step starts to be inhibited, what then happens is that we eventually slow down all of these. We're going to use up this product slowly but surely. As the product is used up, it will unbind from this allosteric site. And as it unbinds from that allosteric site, again, we release that inhibition and it goes back to its normal shape, and the reaction can proceed. So in this way, the end product is regulating how much of it is produced. And this is exactly what happens in the production of ATP. ATP can't be generated and stockpiled. Okay, the amount of ATP that's produced matches the ATP that's being used up because of this idea of end product inhibition. So end product inhibition is one example of regulation of a number of different types that we're going to come across. Here, what we've got is the syllabus statement. And quite a few of these we've touched on when we looked at the standard level stuff. So this idea that metabolic pathways exist, and the metabolic pathway can exist as a chain. Very much in this case, we've got this chain reaction going on here, and we're going to look at examples of that. For example, glycolysis, when we look at 8.2, which is a chain of reactions, all of which are controlled by enzymes. And we're also going to look at certain types of cycles, and we're going to meet Calvin cycle, which is in photosynthesis 8.3, and we're going to look at Krebs cycle, which is within 8.2. So these are going to give us specific examples of how this takes place. But the idea here is to understand that if you've got a series of reactions, all steps are enzyme controlled. They're controlled by different enzymes, and each one of those enzymes can be regulated to speed up or slow down the process, slowing down the process by enzyme inhibition. The idea that enzymes lower the activation energy, we've talked about already when we met the enzymes unit in unit 2. Also, can you make sure that you're clear here about the induced fit model? There's a Sunasamic post. It's an animation with some narration taking place. This covers this syllabus statement very, very well if you need to review it. Then we've got this idea that enzyme inhibitors can be competitive or non-competitive, and I'll explain what that means in more detail in a moment. But competitive means that the inhibitor wants to fit with the active site. So it's competing with the substrate for the active site. Non-competitive inhibition is like end product inhibition that I just showed you, that it's not inhibiting via the active site, it's inhibiting by a different site, often known as the allosteric site. End product inhibition, which I've just touched on and we come back to again, the McGraw-Hill clip, the animation here, which I've posted in the Google Classroom, is a really, really good example. We're going to look then at a specific example, and we're going to look at, again, this idea of databases and looking at how we can look at anti-malarial drugs. We're going to also look at the rates of reaction from experimental data, possibly interpreting graphs and so forth for data analysis. And we need to make sure that we can unpack and distinguish between different types of inhibition from graphical information. This kind of thing does come up in data analysis fairly often. What I'd like you to do in a moment is pause the video, and this task here asks you to think about, with one letter change at a time, to have a series of intermediates that get you from tread all the way to blink, but changing just one letter at a time and showing these intermediate forms. And this is meant to really be analogous to the idea of enzyme pathways, because enzyme pathways are very small steps with numerous intermediate forms. So pause the video there, have a go, see how quick you can be. And here you go, you've got the actual answers which are written down for you. So this is going to be the product at the end of the pathway, and we've got the initial substrate just here. And the initial substrate, it might be that we have many substrates actually coming together in an anabolic process. So remember, don't always think about catabolic processes and breaking things down, but we've also got anabolic processes where we got two substrates or more being bound together to make a larger organic molecule. I've mentioned this already. This is the McGraw-Hill clip. It's also posted in the Google Classroom, and I would pause the video again here and watch this to make sure that you're clear with the terminology. At the same time, I'd also recommend that you look at the Sunasamic narrated animation, and again, make sure that you understand the steps. This slide here is just showing you some examples of pathways versus cycles. So this is glycolysis, and we're going to unpack this a lot, in a lot of detail rather, in 8.2, pardon me. And glycolysis is the first stage of respiration. But what you can see are their intermediate forms, and each one of these is an enzyme catalyzed process. Phosphorylation is a process which undergoes addition or removal of a phosphate group. So we've got an enzyme which is involved in this. Lysis is also when we've got the breakdown of a larger molecule into two smaller molecules. So lysis means breaking or splitting up. Again, it's enzyme controlled. Oxidation, essential that you understand the differences between oxidation and reduction. And remember all the work that we did from previous units. And again, this is showing you that we've got ATP formation. ATP formation is also known as phosphorylation, but we've got a pathway or a chain of reactions going on here. So metabolism is a series of chemical reactions. This is a metabolic chain. By comparison, this one is a metabolic cycle. And what we can see here is Calvin's cycle. Calvin's cycle is an example in photosynthesis. We're also going to look at Krebs cycle. So the details of this we can come back to later on, but there's some links down here as well for you to take a look at. This is a reminder of work that we've done before. And again, the Sunosamid clip, if you've not watched it yet, I would recommend that you have a look. So the idea with enzymes is that they allow you to lower the activation energy. So how do they do this? The enzyme is going to bind with a specific substrate, and the enzyme substrate complex, there's actually a chemical interaction that takes place. It's like a very firm hand grip. That's the analogy that we used earlier on. And the idea here is that puts strain on the bonds, and we need to break and reform bonds in order to make the product. So when we've got an enzyme pathway, we are, by doing induced fit and having this transitional state, we have the impact of lowering the activation energy. And here we can see that overall, we need a little bit of energy to start this process off, but overall, the energy which is of the products at the end here is lower than what we started. So this is energy that is released. Now, be careful, don't call this exothermic, as many of you are chemists. In chemistry, we talk about exothermic reactions, which means that overall heat energy is produced. But when we're looking at chemical pathways within cells, what we want to do is create so-called free energy, energy that allows us to do work. And free energy could be the formation of a phosphate bond, ADP, forming ATP, and then later on, by breaking that bond, we can release energy. So the free energy here is being shown. So this is called an exergonic process. And also what we could have is a reaction where the initial activation energy is here, but then the overall product might end up here. So what we've got is an endergonic process. That means that overall we need to put more energy in to make that reaction take place. Now, in biology, exergonic and exergonic and endergonic processes are usually happening at the same time. So the free energy produced here is then used for another reaction, which might be endergonic. And again, that's another reason why you've got many chemical reactions, this metabolic hub, many chemical reactions going on at the same time consecutively. This is, again, exactly the same image that we've looked at before, and it's reminding you of this idea of the formation of an enzyme substrate complex. This forms a transitional state in terms of the substrate, lowering the activation energy to produce a product. And because the product here is two smaller molecules compared to the larger molecules, this is catalysis. It's a catabolic process. But we also could do this reaction in the opposite direction, and we could take two smaller products and end up with a larger product, which would be anabolic in process. So make sure that you've got those sorts of terms to your fingertips. Within the Google Classroom, you've got access to this PowerPoint, and what I would like you to do is to make sure that you click on each one of these links and just have a little bit of explore. This idea of allosteric enzymes and this idea of feedback inhibition is particularly pertinent to this topic. And an inhibitor is going to have the effect of slowing down the chemical reaction because it's interfering with the shape of the active site and interfering with, therefore, the ability of the active site to bond with a substrate. And just to finish off this first video, I want us to think now about the differences between a competitive inhibitor and a non-competitive inhibitor. So a competitive inhibitor is going to compete with the substrate. So it's going to try and sit in the active site. And if it sits in the active site, that active site is blocked, which means the enzyme can't therefore bond with an enzyme. By contrast, what we've got here is a non-competitive inhibitor. It's binding elsewhere on the enzyme, an allosteric site, but it has the same effect of changing the shape of the active site. So the active site is not capable of therefore binding with the substrate.